Hi biology students. I'm going to finish today talking about types of passive transport. We talked about diffusion, and I know you watched a video a couple days ago that discussed osmosis and the movement of water through a membrane, and we're going to talk in a little bit more detail about that. And then I know the other video you watched was mechanisms of the cell membrane that move things through the cell membrane. And yesterday's video discussed diffusion and how things diffuse through the membrane and why some things can and some things can't. But the topic of discussion today is, how does water regulate in a cell? So think about when you work really hard or work out really hard, you lose a lot of water. So where does that water come from? And then when you drink water, how does that water get into the cells? So next week when you're here, we're going to do a lab on this. So I want to make sure that we really understand the terminology necessary to complete the lab. So osmosis is simply the movement of water molecules through a membrane. And again, it's moving from an area where there's more water to an area where there's less water. So if you get dehydrated when you work out or work hard or sweat a lot, when you get dehydrated, every cell in your body gets dehydrated. And the way that we get that water back in there is by drinking water, but how does it end up back into the cells? So that's what we're going to talk about. Some terms, so an environment that attracts water is called hypertonic. An environment that does not attract water, in fact gives up water, gives water away, we sometimes call it water hating or water fearing, is called hypotonic. And then when that equilibrium has been reached, so isotonic really is equilibrium has been achieved. Equilibrium is achieved and water is equal on both sides of that cell membrane. So if we're talking about cells, it means that water is present in equal quantities or percentages on either side of that cell membrane. So this is why it's important in your body. Just looking at this picture, so this is a red blood cell. These are all red blood cells. The red blood cell on the top is in an area where the water level is higher than it should be. So it is possible to actually drink too much water. So this is the case if you drink way too much water. You have to really force yourself. You're really not going to drink way too much water in ordinary circumstances. But there's been some odd things that have happened. I remember a case years ago where parents used drinking water as a punishment for their child, and she actually ended up dying. Really horrific. This is probably 10 or 15 years ago. I don't remember. Probably 15 years ago. But anyway, so it is possible to drink too much water. This is not a normal red blood cell. This is the normal red blood cell. It's kind of concave in the center, and red blood cells are shaped that way because the nucleus actually dies off after a while, and it becomes that shape. This is a situation where the water level is too low in the solution outside of the cell. So this is, and this is pretty extreme, obviously. But this is when I mentioned earlier that when you get dehydrated, every cell in your body gets dehydrated. Your cells don't work well in that state. If you're ever, prepping for a big exam or something like that, you should always make sure you drink plenty of water the day before and the morning of, because your cells really do work better and communicate better with each other when they're well hydrated. Your body just functions better when you're well hydrated. But anyway, so in this environment, the water level outside of the cell is lower than what's in the cell. So water molecules are being drawn out of the cell, and we get this kind of deflated looking cell. Clearly not a normal cell. And that water will continue to move back and forth until that equilibrium has achieved. Make sure you jot these notes down as we go through them in the videos. These are really important. Obviously, you need them, understand them for the lab, and you also need to know them for assessments like tests and so forth. Want you to have success. All right, so another kind of interesting thing, when we looked at pond water, there were single-celled organisms in there, and you would think like those are a single cell and they're in water that's, you know, it's not pure water, there's other things in there, but clearly the water level is much higher outside of those single-celled organisms than it is inside of them. So there are some mechanisms by which these single-celled organisms actually function to kind of get rid of that, because that water does continue to go in and they need to get rid of it. So they have this really cool mechanism called contractile vacuoles. So this is a single-celled organism called a paramecium, and I'm going to just play this little video clip. Hopefully you can see how that vacuole, that contractile vacuole works. So this is in water, and water is seeping in. Actually, this is the contractile vacuole right there, so that water is seeping in, and you see this vacuole filling up, and then all of a sudden it contracts and it kind of squirts that water out. You can see it getting rid of that water. Here's another close up. There we go. It's crazy, even in a single-celled organism, the amazing functions, the amazing structures and functions that have to be present to survive as a single-celled organism in pond water. So again, you just see that filling up, it's squirting it out. I don't know if that's a water being squirted out. I'm not sure. I'm sure this is on a slide. So the other thing I wanted to show you on this, because we have looked at pond water, there's cilia, little hair-like appendages on the outside of this thing that's it's using for movement. Can you see? Sorry, I don't want all that. But they're wiggling these little hair-like appendages here. I think we can see it better on earlier. And that's how it's moving. So cilia on the outside of this cell for motility. Lots of single-celled organisms actually have that, especially eukaryotic ones, but some bacteria as well. Our bacteria also can have those. But anyway, that's my little contractile vacuole. All right, and then some other terminology that goes along with discussing osmosis and hypertonic and hypotonic. Turgor pressure is something that we refer to when we talk especially about plants. Turgor pressure is said to be achieved when the vacuole is full and that plant cell is kind of plump. So think about what happens with plants. Remember we said the vacuoles in plants are really large. They can take even up to 90% of the cell's volume. So they take up really a lot of space. And we said they're used for water storage, and there's some enzymes in there, and sometimes they store some waste in there. But the primary function really is for that water storage. So when the vacuole is full, the plant is very happy. I maybe shouldn't use the term happy, but that's what the ultimate goal of the plant cell is that vacuole would be full. So think of what happens with plants if the vacuole is not full of water. What do you think the plant would look like? What signs might we see that the vacuole is not full in the plant cell? Well, we would see something called plasmolyses, which we commonly call wilting. So the plant will begin to wilt. That turgor pressure that is very high when the vacuole is full pushes that cell membrane against the cell wall and keeps those plant cells rigid. Okay, but when that's not full, then we have that plasmolyses or wilting. So if you ever have like celery in your fridge at home or carrots and they've gotten a little kind of rubbery and not very appealing, a lot of times you can sort of bring them back to life by just putting them in some water for a few hours, and they might crisp right back up if they haven't sat for too long. Sometimes I have to do that with things in my refrigerator. And I mean, just because they wilt a little bit doesn't mean they're bad and not safe to eat, but it's just not very appealing to eat wilted celery and those kinds of things. All right, and then if in the event that cells take in too much water, there actually is something called cytolyses where they actually can burst. They might reach that point where they absolutely just burst open, kind of a small explosion, and because if that cell becomes very, very full and there still is a higher concentration of water outside of the cell, it still wants to keep diffusing in, and at some point that cell membrane just can't expand anymore. And then I have a short little video clip on facilitated diffusion. That's our last thing we're going to talk about with passive transport. So all these things we've talked about have been passive transport and do not require energy.
A special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier